translation in prokaryotes. Okay, initiation codon is IUG or GUG. Both of them are methionine, but because here in prokaryote we need 4-mil methionine. What? You write it here, 4-mil methionine. This is a 4-mil. F is
4-mil.
And what is the 4-mil? What was the formic acid? HCOOH was formic acid. Yes, without OH this is a 4-mil. Just add to the methionine. But in the eukaryote, just methionine, not 4-mil. This is the first difference between prokaryote and eukaryote. In the eukaryote, the initiation codon is
4-mil?
Is methionine. In eukaryote.
Methionine.
In eukaryote, we have a methionine. In prokaryote, we have a 4-mil methionine.
This is methionine, and this you said this is prokaryote.
Yes, prokaryote is 4-mil methionine. Eukaryote is methionine.
Oh, okay.
Without 4-mil.
Okay. Oh.
Shine-Dalgarno sequence is special for prokaryote. Don't forget. What is the job of that?
It's where the ribosome binds.
Yes, and another thing, why the prokaryote has it? The prokaryote are crazy. They cannot say, please RNA polymerase, please ribosome come here, please everybody come here. Yeah. They need something to shine like a sun and say, I am here. I am a mRNA. The ribosome, please take it off here. Airplane come here. Okay?
It's like a signal.
Like a signal. This is a signal sequence. What is Shine-Dalgarno? It's shiny.
Yeah, shiny.
And said the ribosome, please bind to me.
Yeah.
We need the initiation factor. We need the 30S initiation complex. And you understand what is the 30S? It has a mRNA.
Yes.
It has a 4-mil methionine tRNA. It has an initiation factor. And the 30S subunit for sure.
Yes.
70S initiation complex is 30S plus 50S. Plus 50S, this is a
together
70S. Okay, we need the P side. F methionine tRNA comes here, and the A side is empty. This is a position of A and P in the initiation. A and P is different in elongation and termination. Now, how should we remember? I remember it like that. This is here. We have 3 subunits. Exit, P, and A. Again, exit, P, A, EPA. First of all, the 4-mil methionine comes to A or P, comes here in the initiation. And then the next amino acid comes here. It bonds to the methionine. Then this is the mRNA. mRNA is fixed, but the ribosome can transfer like that.
Yes.
Again, this is a mRNA and I am a ribosome. The ribosome transfer through that, and in this case, the P comes to the 4-mil methionine instead of subunit P comes to the exit.
Yes.
And it's go away. The next one comes to this.
So when one amino acid comes to P,
for example, this is
E?
This is nothing here, okay? When it's go away, the methionine comes here. This is a methionine now.
Yeah.
And the next amino acid comes to this one, a star, for example. And the next one, the moon, comes here.
But it starts at E?
A start?
No.
It exit at E.
Exit,
E is the exit side.
Yes. But you said that it first comes to P site, right?
Yes.
And then the next one, it comes to A site.
The next amino acid, yes.
Yes.
But when the ribosome move, the methionine comes to exit.
Yeah.
And the next one comes to P. And the new one comes to A again.
Yeah.
And then we elongate. Okay. Now, what's happened here? We have amino acid tRNA binding in elongation with the help of the elongation factor 2 and GTP to the 8 side. Peptide bond formation by the important enzyme peptidyl transferase or ribozyme. What is the ribozyme?
It's RNA enzyme.
Yes, all of the enzyme are 99% is peptide. Just one enzyme is RNA, and the name of that is ribozyme. Do you remember what type of enzyme it was? Catalytic enzyme.
Yeah, it is.
Do you remember the shape of that?
Yeah, it cleaves like.
It was somewhere here in the fist.
Yeah, it can cleave like the mRNA, can't it?
It can cleave the peptide, peptidyl No, it's not cleave. It make a peptide bond. The job is peptide bond formation. This is this line. This is the shape of that. For example,
Like after
and this
KO
is here.
mutation.
You see?
I need to show you that.
This is here.
After KO mutations or something, it's here. It's KO mutation, KO mutation.
Before.
Before, yeah, it's before, right?
It should be somewhere here.
Inhibition. This thing.
The ribozyme.
That's what I was talking about.
Cleavage of mRNA, but now we are in the ribosome,
Oh.
and the job of that is a peptide bond formation, and peptide bond formation.
In the ribosomes, right? In
Yeah.
ribosomes.
Here in a on the big subunit of ribosome, big subunit of ribosome.
So it makes the peptide bond.
Yes.
Okay.
But here, yes, mRNA degradation is one of the job, and actually it do it here also when the when the transcription is finished.
Okay.
Translocation with the help of elongation factor G happens and GTP. Now I wanna ask you a question: how many GTP is consumed until now? One GTP here consumed, two bonds to initiator factor. The second mole of GTP is consumed here to bring the tRNA amino acid tRNA to bring it. The third GTP is used here for the translocation.
What is a translocation?
Translocation is here.
Okay, okay.
It needs the third GTP come here, and then it's take it. Elongation factor GTP, take it. Move one codon. Again, one GTP for other codon.
Okay, so for each codons, it has to like move. It needs a GTP.
Yes. No,
Okay,
the
okay.
teacher in this topic give you this example. Said, I have a 50 codon. How many GTP use? What do you think? From this 50, for sure, one of them is initiation, 48 is between, and one is the final codon, yes, termination.
Yeah.
For initiation, you need one GTP. For elongation, for one amino acid, you need two. One GTP, two, second GTP, okay?
Yeah.
It means 48 multiplied by
Two.
2, this is at 80, and 16 is 96. Now, for the termination, stop codon releasing factor. Do you see any GTP here? Nothing. 96 plus 1 together is 97 GTP consumed. I hope you understand it.
Yeah.
Again, how many GTP here on the elongation? 1, 2.
Two.
But for initiation, just 1.
Yeah.
And you should separate initiation by that. One of my friends said, okay, we have 50 multiplied by 3. No.
Oh no.
Understand?
But the last one, is that for termination?
For termination, the stop codon doesn't need any
any.
energy
Yeah.
because we don't want to move much more.
Yeah.
And releasing factor, yes, the factor is released. Everybody go home.
Yeah.
Understand
Yes.
until here?
I do.
What is the other name of peptidyl transferase? Ribozyme.
Ribosome. Yeah, you just.
Ribozyme. It's make a peptide bond.
Yes.
No, the prokaryote story is finished. No, unique feature of eukaryotic translation, everything is same. Just we have a unique formation. First of all, here we don't have Shine-Dalgarno. No Shine-Dalgarno here. No Shine-Dalgar. No, and you know this is from the MRT, yes?
Yeah.
5' cap, we have ribosome binding, but we don't have a cap of what? Of
What?
prokaryote. The prokaryote doesn't have
No,
cap, but the cap is
eukaryote.
unique feature of eukaryote.
Yeah.
Ribosome. Free ribosome, protein of cytosol, nucleus, mitochondria. Bond ribosome is secretory protein of endoplasmic reticulum, Golgi, lysosome, cell membrane. If the ribosome is free, this protein is just for the consumption of cytosol, nucleus, mitochondria. But if the ribosome is bound, these are secretory protein. What does it mean, secretory protein? See, this story. حداقل درو ببند.
They secrete something or
You
what?
see, This ribosome,
Yeah.
it secretes and get it to Golgi, and
Yes.
Golgi secrete them. This is a bond ribosome, okay?
Yeah.
This is a bond ribosome.
Membrane boundary.
Now back here. General feature of translation, the direction is always in eukaryote, prokaryote doesn't different, 5 to 3 from above to low, N to C. I always said 5 and 3C. 3C. And this is C. I don't know how. You don't know anything in Persian?
No.
We pronounce it 3C, 3, 3, 3C, 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 3C.
Oh, yeah.
3C,
Yeah.
5N. Understand
I know.
it?
Yeah, you talked about the alphabetic, right?
Yes, alphabetic.
Yeah.
3C, and 5'
Yeah.
N.
Yeah.
Polysome are formed. Polysome. We have here prokaryote, eukaryote is a general feature.
Yes.
One polypeptide chain for ribosome. It means the ribosome, every ribosome can produce one polypeptide chain, not more. Require 4 high energy bond, peptide bond, ATP to AMP and 2 GTP to GTP. That's very important. 4 high energy bond. ATP to AMP, how many bonds break?
1.
2.
How come?
3-phosphate to monophosphate. You see, ATP should produce ADP. ADP should produce AMP. Yes.
Oh, yeah.
2 bond and
2.
GTP to GTP. GTP to GTP just one bond.
One.
But because it's here 2, it means 2 and 2, 4. 4 high energy bond require to produce one peptide bond. Okay, now
In the MRTs, it said it's 4 needed to incorporate 1 amino acid.
yes, 1 amino acid peptide bond between 2 amino acid actually. No, if it's I ask you 50 codon, how many GTP? I want to ask you now how many high energy bond. You should say how many peptide bond do you have between 50 codon?
How many peptide bond I have?
Yes. If you have a 50 codon, how many of them is amino acid? 49 minus 1. Why? Because one of them is codon. It's a stop codon. Stop codon, it doesn't have any amino acid. If you have a 49 amino acid, how many peptide bond do you have? You have a 5 finger, yes? How many bonds you have? 1, 2, 3, 4.
4.
Always minus 1. Okay, always minus
1.
Okay, 49 minus 1, you have a 48 peptide bond. Now, if one peptide bond needs 4 high energy bond, it should multiply it by 4. And the result is something.
But why do we minus it with 4? I mean, with 1? Because
1, because here is a stop codon, the first one.
Yes, and
the next one to make a bond.
Okay.
I have, you see, for example, I have a 2 subunit, just 1 bond. 3 subunit, 2 bond. Always less than 1.
Okay. I understand.
And this is multiplied by 4. And other thing, inhibitor of protein synthesis is the antibiotic, chloramphenicol, erythromycin, tetracycline, streptomycin, puromycin. The name of them is not important. The job of them is important. If you want to inhibit the protein synthesis, you can inhibit peptidyl transferase. You can inhibit translocation. What does it mean, inhibit translocation? It means inhibition of elongation factor GGTP, because it caused the translocation, it caused the movement. If you can stop and inhibit amino acid tRNA binding by the help of tetracycline, you inhibit protein synthesis. What was the job of amino acid tRNA binding? Amino acid tRNA binding with the help of elongation factor 2. It means you inhibit elongation factor 2 GTP. Understand? This is a 2, TU, not number 2, okay? 2. And maybe you inhibit 30S subunit. In this case, the 30S complex doesn't start, that this story is completely stopped. And if you cause the early termination, what does it mean? It means you change the codon.
Yeah.
Do you remember RNA editing? RNA editing in ApoB100, ApoB48?
Yeah.
This is the RNA editing, but this is the early termination. I call the early elimination. It means early elimination. Sometimes it's good, sometimes it's bad. It depends where.
Okay.
The place is important. The operand model, you know it, yeah?
Yeah, I don't need to go through this one.
Good. Just I have one question.
Yeah.
If I have a glucose, if I have a lactose, the lac operon is done or not?
If you have a
If I have a glucose in my body, if I have a lactose in my body, and I am a bacteria, okay?
Yeah.
The lac operon is a start in my body or not?
you have to have a lactose. You cannot.
I have a glucose, I have a lactose.
No,
Why?
because you already have it.
Yes.
Yeah.
If I don't have a glucose and I don't have a lactose and I don't have a energy, what's happened? If the lac operon is done?
No,
Why?
because you don't have the energy, right?
You see, I don't have any energy. The lac
Yeah.
operon wants to compensate my shortage of energy, yes?
Yeah.
No, I don't have glucose, neither I don't have lactose also.
Yeah.
No, the lac operon should work or not.
It should work in order to make it, right?
Yes, but I don't have lactose. And
Yeah.
what is the job of the lac operon?
It's
Lac
to
operon?
it's to make a lactose
No, make a
So
lactase.
it's the lactase. It's to make it make the enzyme to break down lactose, right?
Yes.
So yeah,
The name of this enzyme is lactase. And
Lactase.
what is the lactose?
lactose is a sugar.
It's a disaccharide.
Sugar.
Which monosaccharide? What is the monosaccharide of lactose?
Galactose and glucose.
Perfect. Mechanism of gene regulation in eukaryote?
Yeah.
Really? You wanna go through them
I
now?
don't know. Just is it important?
Yes,
what is
for
it?
sure. All of them are very,
Just go
very...
superficially through it. Just but if it's just point out what's important because I've sort of read it through. You had
The Gordon experiment.
Yeah.
This is the oocyte.
Yeah.
Without nucleus. This is the enucleated. The oocyte has a haploid. Haploid, do you know what is the haploid?
Yeah.
Just one N. But the tadpole or small intestine and epithelial cell, micronucleus nucleus of the epithelium, this is a 2N. Instead of fertilization by sperm, I enucleated and I inject by microinjection 2N cell to the oocyte. What's happened? Three fate. No cell division. Blastula, abnormal embryo, normal embryo, and normal frog forms.
Yeah.
This is a Gordon experiment. Gordon enucleated frog oocyte, microinjected the nuclei of the tadpole intestine cells. Some develop into frog. Genome equivalence, housekeeping gene, tissue-specific gene. What's happened then? How many type of cell do you know? Somatic, germ. Somatic cell, germ.
Is there any body cells, germ cells?
Yes. What is the difference between somatic cell and germ cell?
Germ.
No difference. The material, the gene is equivalence.
Okay.
Both of them are 2N, 46 chromosomes. Or in this case, why one of the cell is nail, one of the cell is hair, one of the cell is eye? Why? Because they are differentiated differently.
Yeah,
It means the other gene is silent. Just one of such a gene is express itself. I want to ask you a question. The germ cell could be differentiated to any type of cell? Yes.
Germ cell.
Germ cell, like a bone marrow.
Yeah.
It could be differentiated to any type of cell. The somatic cell,
No.
we think no, but they do it. The Gordon proof
Oh.
that the somatic cell from intestine, the one cell from intestine, the nucleus of that injected to oocyte and new frog form. It means such a gene, housekeeping gene and tissue specific gene is expressed here and the other one exists, but it's silent.
Yeah.
What is the blastula? Blastula is synonym with blastocyte. What is the blastocyte?
It's like a what do you call it? It's an embryo that has been divided.
How many times?
I don't know.
5 times.
Okay.
2 to the power 5. It means 32 cell.
Okay.
In this case, it could be implanted into the uterine in the endometrium.
Yes.
Implanted happens. Nuclear transplantation in mammals by the dolly and lamb. You know how the dolly produce?
It was a cloning or something.
That is a cloning story, yes. Enucleated oocyte transfer the nucleus of the mature other cell. Reproductive and therapeutic cloning happens. Now, we should understand the three fate here. Sperm and oocyte fertilization, zygote, blastocyte, normal mouse. If I get one of the somatic cell of adult mouse, I get the nucleus of that and I inject it to enucleated oocyte, and I produce a blastocyte, is a cloned mouse.
That was the dolly, right?
Yes, and this Gordon also. And if I do the same, but I don't want to produce a new animal, I just want to produce new tissue. I want to produce muscle cell, neuron, blood cell. It's happened. Somebody get the neuron degeneration, multiple sclerosis. Okay, I do it. Somebody has a cancer. I make a blood cell for that by this method. Or maybe somebody lose the muscle on the burn accident. But these methods are very, very expensive and a lot of research done.
Okay.
Over them. Level of gene regulation in eukaryote. No, it's finished. The topic is until here.
Okay.
Do you get the mechanism of gene regulation in eukaryote?
Yeah,
It's a mechanism, not the level. These are two topic or one topic? Regulation of mRNA.
What was that?
Cloning by nuclear transplantation. It was this 47.
Number
You
47.
can write topic 47.
It's page number
93. And regulation of mRNA synthesis and processing in eukaryote is this one. In eukaryotic. Again in eukaryote. For sure, everything is done in eukaryote, not in the prokaryote, okay? Because the prokaryote is simple. I finish for you this and this also. And then we can postpone the other.
Okay.